Okay, good morning, everyone, and welcome to the 2021 Gairdner Lectures, Minds That Matter. My name is Rick Wozniak, and I'm a professor in the Department of Cell Biology in the Faculty of Medicine and Dentistry. I've worked in various capacities with the Gairdner Foundation over the last decade or so, and today I will function as the MC for the Gairdner Foundation Lectures. Before we get started, I would like to first acknowledge that the University of Alberta is located on Treaty 6 territory. We respect the histories, languages, and cultures of First Nation, Métis, Inuit, and all First Peoples of Canada, whose presence continues to enrich our vibrant community. As many of you are aware, the Gairdner Foundation Lectures are an important annual event at the University of Alberta, and many universities across Canada this last week in October. While we have had a change in format to the symposium the last 2 years, we are happy to continue to bring this event to you virtually today, and we hope that everyone enjoys it. Today's events will include the morning's public seminars by 2 Gairdner laureates, and then in the afternoon, a question and answer session between the laureates and registered trainees. I would like to start by thanking various individuals who to putting this event together. First, we thank the Gairdner Foundation, in particular, Janet Rossat, President and Scientific Director of the Gairdner Foundation, Paige Johnson, the Manager and Fund Development Communications person there, for their assistance in making this day possible. I would like to also thank various members of the University of Alberta Faculty of Medicine and Dentistry for their contributions and assistance in putting this event together, including Dean Hemelgarn, Dr. Richard Lenner, the Vice Dean of Research, Dr. Greg Funk, the Associate Dean, Research Graduate Programs. I would also like to thank Drs. Christy Baker and Tom Hobman, who you'll hear from in a minute as they will provide introductions for today's speakers. Importantly, I would also like to thank Lise Hetu, the Assistant Director of Alumni Relationships in the faculty. She is a critical figure in overseeing today's events, and wanted to send out a real thank you to her for keeping this event running smoothly. Finally, I would like to stress the importance of financial contributions from the Gairdner Foundation, the governments of Canada and Alberta, the Canadian Institutes of Health Research, and the Globe and Mail for supporting today's events, and they're indicated on the bottom of the slide you're seeing. At this point, we'd like to invite Janet Rossat, Scientific Director of the Gairdner Foundation, to say a few words about the foundation and the importance of its work that it does. Unfortunately, Janet was not able to join us live today, but she sent along a recorded message that we'll play for you now. So Elise, if you could take care of that.
Hello, and welcome to today's Gairdner National Lecture. I'm Janet Rossat, President and Scientific Director of the Gairdner Foundation. At Gairdner, we're best known for the prestigious Canada Gairdner Awards, which we've been awarding for over 60 years. 395 awards have been made, 97 laureates have gone on to receive the Nobel Prize, including this year with Dr. David Julius. We're delighted to be able to bring our laureates to speak at universities across the country, where they inspire us all to greater heights in our own research. Our laureates this year have made discoveries that have had an impact on millions of lives around the world, from diabetes to breast and ovarian cancer to zoonotic diseases. The reach of their research is truly global. Your Gairdner National Lecture today is part of a series of virtual events happening from coast to coast as part of Gairdner Science Week. Please visit our website at gairdner.org or check out our social channels at Gairdner Awards to see the full schedule and register to be part of an exciting week of outstanding science. I'd like to thank our sponsors, the Government of Canada, CIHR, the governments of Quebec and Alberta, and the Globe and Mail for their ongoing support of our programs, as well as other sponsors listed on our website. Their support makes events like this possible. I hope you enjoy today's talk and that you'll join us for all the other fantastic programming that Gairdner Science Week has to offer. Thanks and enjoy.
Okay, thank you very much, Janet, if you're out there somewhere listening. It's a great pleasure this year to welcome to our program two Gairdner laureates, Dr. Roel Nusse from Stanford University and recipient of the 2020 Canada Gairdner International Award, and Dr. Malik Paris from the University of Hong Kong and recipient of the John Dirks Canada Gairdner Global Health Award in 2021. Our program will include a 40-minute presentation by each laureate, followed by a Q&A session, about 10 minutes. During the Q&A, if you have questions, please raise your hand electronically in the bottom of the presentation screen that you see. Once I've recognized you as asking a question, you'll be unmuted, sorry, and you can go ahead and ask your question. You can also ask questions through the question and answer chat box, and we'll try to work these together as best we can to get as many questions as we can to the speakers during the amount of time that we have for questions. So I'd like to start our afternoon, or excuse me, our morning events here by introducing Dr. Christy Baker, assistant professor in the Department of Oncology, and she will do the introductions for Dr. Nusse. Christy.
Thank you so much, Dr. Wozniak. So it is my great pleasure today to introduce Dr. Roel Nusse. He is the professor and chair in the Department of Developmental Biology at Stanford University School of Medicine. He is also the Virginia and Daniel K. Ludwig Professor of Cancer, and he's also an investigator for the Howard Hughes Medical Institute. Now, Dr. Nusse completed his graduate studies in the Netherlands Cancer Institute in the University of Amsterdam, where he worked on the mouse mammary tumor virus proteins. He then began a postdoctoral fellowship at the University of California, San Francisco, with the very renowned Dr. Harold Varmus. And there he worked on studying the mouse mammary tumor virus induction of cancer, where he was really working on looking at proviral integration upstream of a proto-oncogene that was later identified as the homolog for Wnt. He began his own independent research career in 1982 at the Netherlands Cancer Institute. And then in 1990, he was stolen back by Stanford University, where he became an associate professor at the Department of Developmental Biology. And since then, he has served as the chair of this department for 3 terms, starting since his first appointment. Now, Dr. Nusse has been awarded the Canada Gairdner International Award 2020 for his pioneering work on the Wnt signaling pathway and its importance in development and cancer stem cells. Dr. Nusse made the original discovery of the Wnt proteins, along with Dr. Varmus, and this was published in a little journal we may all have heard of, known as Cell. And in combination with his demonstration that this mouse mammary tumor virus could induce oncogenic transformation, he also showed that the Drosophila homolog of Wnt, which is known as wingless, was a key developmental gene. And the real significance of both of these discoveries together is that it really led to the realization that cancer and normal developmental pathways are very tightly linked. And this is something that many of us who work on cancer, it's a standard in our research today. Now, he didn't stop there. Dr. Nusse went on to discover most of the many other significant Wnt signaling pathway mediators, including beta-catenin, which relays the signal from the receptors into the nucleus, in addition to the receptors themselves, which are known as the frizzled family receptors. Now, I am a researcher who studies colorectal cancer, and so the very, very first pathway I learned as I was studying the adenoma to carcinoma transformation sequence was actually the Wnt pathway. So this is a project and a topic that is near and dear to my heart. And Dr. Nusse has been recognized in the past for the amazingness of his discovery and the significance of it when he won the Breakthrough Prize in Life Sciences, which is funded by a few people we may have heard of, known as Mark Zuckerberg and Sergey Brin, in 2017. And this is a prize that is given for discoveries that extend human life. So I'd like to present to you today Dr. Nusse for his talk on From Cells to Tissues, Wnt Signaling as a Common Organizer of Growth.
Thank you, Christy, very much for this introduction and overview of my work. My presentation today will indeed be dealing with the Wnt signaling pathway mostly, and how it is implicated in how tissues grow and how cells divide. And I'll start by sharing my screen. So this is the title of my talk, From Cells to Tissues, Wnt signaling as a Common Organizer of Growth. And this is indeed a issue that goes to the heart of normal development and how multicellular organisms, how they grow during embryogenesis and how they maintain tissues later in life, but it also goes to the heart of cancer. And I'll try to make links between Wnt signaling and normal development and tissue homeostasis and cancer during my talk. But I want to start with a view of how tissues and embryos grow from the point of view of evolution, where we can start with unicellular organisms, microbes that can live in the ocean or in the soil, where sometimes these cells may undergo some kind of differentiation, but in principle, every cell, every single cell organism can proliferate. But during the course of evolution, when the Metazoan kingdom arose, and that ultimately led to the different branches of the kingdom into sponges, the porifera, Cnidaria, these are animals that live in the ocean, Ecdysozoa, insects mostly, and then also the chordates, including vertebrates such as mice and also ourselves. What actually arose then is that not every cell in the tissue would proliferate. There were unequal rates of cell division. And that is already manifested early in development. And whether it is the development of a sponge or a sea anemone or an insect or a mouse, what actually occurs early in development is that there is a local growth zone. And that local growth zone, indicated here when the embryo is still just an undifferentiated ball of cells, will actually lead to the development of an axis of asymmetry, where the developing embryo starts to differentiate into an anterior and to a posterior domain. And it is that posterior domain that keeps on growing while the anterior domain slows down the rate of proliferation. And you can imagine that if you have a ball of cells where there's just a minor population of cells maintaining their proliferative status, that you get the elongation of the developing embryo, and something that ultimately will develop into an anterior posterior embryo, with the anterior domain developing into the head and the posterior domain into the tail of the embryo. And quite remarkably, that sort of organization of a growing embryo is actually conserved all the way across the Metazoan kingdom. It has also been established that local growth is induced by signals. And those signals, they act in a very local way. Those signals actually emanate from what is going to be the posterior domain of the developing embryo, and they set into motion the proliferation of adjacent cells. Now, what are those signals? And among those signals, the Wnt proteins, indeed proteins that we have been working on for many years, are quite prevalent. Wnt proteins are secreted factors. The proteins that start with a signal sequence. They are highly conserved in evolution, and in particular, the pattern of the cysteine residues in a Wnt protein is conserved in a Wnt protein, whether you actually look at a Wnt protein in a mammalian organism or a Wnt that is found in more simple organisms such as hydra and sponges. Now, in addition to that highly conserved pattern of cysteines, there's another property of the Wnt protein that is very important and also conserved, and that's that Wnt proteins are modified by a lipid. So these are actually proteins that, even though they are secreted from a cell and indeed signal to an adjacent cell, are actually very much tightly bound to the membrane of the cell through that palmitoylate. And this is very important in early development, where quite often you start, of course, with an embryo that only contains a few cells. And it has been shown in various organisms that even in those embryos consisting of just a few cells, one cell can actually make a Wnt, and the adjacent cell is going to respond to the Wnt and will be induced by the Wnt to proliferate and ultimately to adopt a particular fate. So Wnt proteins, through the properties of being modified by a lipid, being secreted from one cell and acting on adjacent cells, is very important for organizing early embryos throughout the animal kingdom. Now, it is indeed quite striking in that context. If you start to look at where Wnt genes have actually been found, you don't see them in the unicellular organisms. And indeed, unicellular organisms, they wouldn't need the Wnt anyway, because every cell can divide. They don't need some kind of multicellular organization. But if you now start to look across the evolutionary development of the different branches of the animal kingdom, you see that the origin of Wnt genes is indeed found right at the earliest branch of the metazoans. Sponges have Wnt genes, and so do sea anemones, and so do insects, and so do vertebrates. Now, what is also quite striking is that if you start looking now where Wnt genes are expressed, you see that throughout the animal kingdom, whether these are sponges or mice, there's an early domain of Wnt expression. And that early domain of Wnt expression is indicated here by these red circles. And here you see some examples of real developing embryos. This is a Gnaearia embryo, so that ultimately is going to form a polyp. Or this is a chordate, a developing mouse embryo. And you see that there's a very high local expression of a Wnt gene, and this is what's going to form the posterior end of the developing embryo. And this is here a striking expression of a Wnt gene at the tail bud of a developing mouse. So Wnt genes are indeed among those signals that are setting up a local growth zone. And by doing so, they actually set up the primary axis, the primary anterior-posterior axis of a developing animal. And again, that is conserved all the way across the animal kingdom. Now, it is not only in developing embryos, but also if you now start to look at tissues, and these could be tissues present in vertebrates, you see that there are local signals that are responsible for the proliferation of cells. And those local signals, they act on what we call cell progenitors. Those cell progenitors are activated by a local signal coming from nearby cells. And those progenitors, under the control of these local signals, will start to divide and then generate their daughter cells. Now, these local signals, again, they make sure that only the progenitors are going to be activated by the signal, but the differentiated daughter cells coming from the progenitors, they leave what is called the niche where these signals are active. They don't see the signal anymore, and consequently, they lose their proliferative status. And that is how you organize many different tissues into a population of dividing progenitor cells and non-dividing differentiated cells. And again, that is something that you find in many tissues, including in the intestine that Christy is studying, but also in tissues like the liver and also in the skin. And among those local signals are again, the Wnt proteins are very prominent. They are made by cells that are adjacent to the progenitors. The progenitors will proliferate. They will start to generate more differentiated daughter cells. Those daughter cells are now not dividing anymore, and they execute the function of the tissue. Now, on top of that, these differentiated daughter cells, they are not only losing the contact with the cell proliferative signal, they also become arrested in the cell cycle through the expression of inhibitor genes that are actively prohibit cells from dividing. And this is very important for normal tissues. Actually, the vast majority of cells in our tissues, they are not proliferating. It is only a small population of these progenitor cells that are dividing. And that also has implications for the likelihood that in a tissue, you're going to develop cancer cells. It is now quite prominent to find that cancer cells arise from these dividing progenitor cells, and that the non-dividing differentiated cells in a tissue, by having lost the ability to proliferate, are much less likely to turn into a cancer cell. So these are the themes of my presentation today. I'm going to tell you more about how these local signals, including Wnt proteins, are instrumental in keeping progenitors in a proliferative state. But I'm also going to tell you how it is possible that differentiated cells in a tissue, and it will be mostly focused on the liver, are actually arrested in the cell cycle through an inhibitor activity. So let me first tell you more about the Wnt protein and the Wnt sickling pathway, how that all operates. Some 10 years ago, a colleague of mine at Stanford, Chris Garcia and Claudia Janda, were actually able to determine the structure of a Wnt protein. And what they found is actually that the Wnt protein indeed contains that lipid modification, a palmitoylate, and quite strikingly, that palmitoylate sticks out of the remainder of the Wnt molecule. That was unexpected, but what was subsequently found is that that palmitoylate sticking out of the Wnt protein is instrumental in binding of the Wnt protein to one of its receptors, and the receptor molecule is called frizzled. Now, on the other hand, when the Wnt protein is not yet bound to frizzled, to its receptor, it can actually interact with a membrane of a cell, also through the palmitoylate. And again, that makes sure that the modified Wnt with the palmitoylate is acting as a membrane-bound protein, acting over a very short distance in a tissue. A little bit more about the Wnt sickling activity. I'm displaying it now in a state of a cell that is not yet activated by Wnt. There are two kinds of receptors on the cell surface. One of them, again, is frizzled, and the other one is a co-receptor called LRP. Now, in that cell, again, it is not activated yet by Wnt. There's this protein called beta-catenin, and the beta-catenin protein is held in an off state by the interaction with a number of other proteins, including axin and APC, and then a protein kinase called GSK3. In that complex, the beta-catenin protein is going to be phosphorylated by the GSK3 kinase, and it leads to the destruction of the beta-catenin protein. And of course, the protein that is being destroyed cannot be active at all. However, when the Wnt protein is now produced by an adjacent cell, and it's going to bind to the receptors, to frizzled and LRP, there is a rearrangement of that complex in the target cell that will actually lead to the release of the beta-catenin protein from APC and actin and GSK3. Beta-catenin is now not phosphorylated anymore, and it accumulates in cell and will now go to the nucleus of the cell, where it will partner with a DNA binding protein called TCF. And that configuration of TCF and beta-catenin, together with many other nuclear proteins, will lead to the activation of Wnt target genes. There's a large number of Wnt target genes, and if you start to compare different Wnt targets between different cell types, you find that almost any cell type has a unique pattern of Wnt target gene expression. And Wnt sickling is implicated in the growth of many different tissues, so it makes sense that different tissues have their own set of Wnt target genes. However, there are a number of exceptions to the pleiotropy of the Wnt target genes. And these are genes that are actually implicated in Wnt sickling itself. One of them is the gene called AXIN2. It is activated by Wnt in many different cells and many different tissues, and can therefore lead to identifying where Wnt sickling is active in a cell, because of you can look at the expression of AXIN2. AXIN2 is a homologue of AXIN, and it actually can participate in the destruction complex. It is actually a negative feedback regulator of Wnt sickling. There's another Wnt target gene called LGR5. And LGR5 is a receptor molecule. It will go to the membrane of the cell, and there it can interact with a protein called arspondin. And arspondin is a molecule that has been identified some years after the original identification of Wnt as a so-called Wnt cofactor. The combination of a Wnt and arspondin, again, also secreted factors, can actually amplify the Wnt sickling by arspondin binding to the LGR5 receptor. And LGR5 is also among the more universal Wnt target genes. Now, many of you may have heard about arspondin and LGR5 because of the ability of researchers to grow tissues and stem cells in the dish in so-called organoids. And it has been shown now by many different labs that you can actually take out of tissues like the mammary glands and the lung and the stomach and the prostate, the stem cells from those tissues, the progenitor cells, and amplify them in the dish into structures that resemble the tissue where they come from. So a liver organoid starts to resemble the normal liver, and a stomach organoid resembles the normal organization of the stomach. And the list of organoids that are now grown in the dish is growing quite rapidly. Now, those organoids can only be maintained in a state where the cells can proliferate and differentiate by adding either active Wnt protein or a combination of active Wnt and arspondin. And it is now that quite powerful combination of arspondin and Wnt that has actually led to the propagation of these organoids in cell culture. This can also be done now from human tissues, and it opens up all kinds of interesting possibilities to study the proliferation and the organization of human tissues in culture. Wnt sickling can also be studied quite conveniently now in tissues by the use of the genes that I indicated earlier, LGR5 and AXIN2, because what researchers have been able to build are so-called reporters and lineage tracing tools, where you take now the promoter of the AXIN2 gene, a Wnt target, and you drive now the expression of a reporter where the LAC-C gene is activated by AXIN2. In other words, because AXIN2 is responsive to the Wnt signal, you can actually now monitor Wnt sickling in tissues, and this is now shown here in the liver. I'll come back to that later in my talk, where you see that there are cells that are turning blue. Those cells have been activated by Wnt. They are adjacent to a blood vessel in the liver called the central vein. But also the H5n2 promoter can be used to drive a transcription factor RTTA, and then H2B-GFP in an indirect way, activating H2B-GFP where Wnt sickling has been active. This is actually a construct that was developed by Elaine Fuchs, who was one of the other recipients of the Gairdner Award in 2020. And Hans Clevers, a researcher in the Netherlands, has used the LGR5 gene, driving the Cre recombinase to also lead to GFP expression, monitoring Wnt sickling in vivo. And it is indeed also now by the use of these lineage tracing tools that people can monitor in vivo, so in intact tissues, whether certain cells are proliferative. You can induce now the presence of so-called clones, labeled clones, in a tissue like, this could be the skin, and you can then watch by using imaging of the proliferative cells that these cells are then able to generate clones, including the original progenitor cell, but also the more differentiated cells in the tissue. And that has led also to remarkable results where you can see that there are now stem cells present in a tissue like the skin. These are present in the basal layer of the skin tissue. And over time, those labeled cells are giving rise to the differentiated cells in the skin, including the flattened keratinocytes. In the intestine, tissue that Christy is working on, it has been shown that there are stem cells, progenitor cells in the base of the crypt of the tissue, and over time, those labeled cells will give rise to the differentiated cells that are ending up in the villi of the intestine, but also in the mammary gland and in the brain. There's now ample evidence that there are progenitor cells and stem cells that are activated by Wnt that proliferate and over time give rise to the differentiated cell of the tissue. And here to the left is a list of tissues where you can find progenitor cells and stem cells that can be traced by the presence of the LGR5 gene or the LGR5 protein and H5n2. So that leads now to the conclusion that Wnt sickling is causing cells to divide. And this is very important. We take it for granted that Wnt is a proliferative signal, but it turns out that we actually don't know much about how that actually works. And this has been bothering us for all the years that we have been working on Wnt sickling. How is it possible that Wnt is actually leading to cell proliferation? And I want to now illustrate our recent findings that address that issue by going back to the organization of cell division and the cell cycle, where cell division is starting by cells that are present in a resting state, in G1 state, entering S phase, where the DNA is going to be replicated, then entering another intermediate state called G2, and then cells are going to enter mitosis, and that is actually where cells are going to divide. So Wnt sickling is indeed causing cells to divide. How does it work, and how does it work on the cell cycle? And I'm going to tell you now about the experiments that we have been doing in the liver, where the liver as a main organ is subdivided into liver lobes. These are the large lobes of the liver. But then within the lobes of the liver, there are these repeated units of cells that are called liver lobules. And the main cell type of the liver, this is an hepatocyte, is organized in these liver lobules surrounded by blood vessels called the portal vein. The portal vein is where actually the foodstuff that is produced in the intestine of the tissue is going to enter the liver, and that is where the food is being processed. But then in the middle of a liver lobule is another blood vessel called the central vein. And the central vein is essential for the organization of these liver lobules in these typical hexagonal patterns of organized cells. It has been shown by many different labs that Wnt sickling is actually necessary for the postnatal growth of the liver. So the liver is one of the tissues where the expansion of the tissue is, and the replication of cells is early on in mouse development dependent on Wnt. Wnt is a growth factor for liver expansion. Now we wondered how that would work, and we found that if you start simply to look at where Wnt are expressed by in situ hybridization, quite strikingly, we see that the Wnt is actually made by the endothelial cells of the blood vessel, the central vein, which is present in the middle of these liver lobules. And you see by in situ hybridization here that the very thin endothelial cells, so these are the cells that make up the blood vessel, they produce high levels of the Wnt RNA. And what you can also see is that if you now start to look at a Wnt target gene, and this is a gene called Tbx3, which is going to be playing a major role in my subsequent experiments that I'm going to show you, that Tbx3 is expressed in a sort of a gradient of the hepatocytes that are adjacent to the central vein, to the endothelial cells of the central vein. So the central vein makes a Wnt. The adjacent hepatocytes, they start to activate a Wnt target gene. And that Wnt target gene is a gene called Tbx3. There are many other Wnt targets that are activated by Wnt in a pattern similar to Tbx3. Those could be target genes including enzymes that are important for the liver function. But the Tbx3 gene is actually instrumental in the growth of the liver lobule. And this is what we found, and I'll explain later how it actually works in more detail. But I'm first going to point out that the Tbx3 gene, again, it is a Wnt target in the liver, is producing a DNA binding protein. It is a transcription factor. But surprisingly, it is acting as a repressor of the expression of yet other genes. So let it sink in for a little bit. So the Wnt signal, it leads to what we call a zonated pattern of a repressor protein. And that brings up, of course, an interesting question. How can a Wnt, through the Tbx3 protein, regulate cell division and cell proliferation? How can it be that a repressor is important for the proliferation of target cells? So we wondered, finding that Tbx3 is activated by Wnt, and knowing that Tbx3 is a repressor, what are actually the targets of the Tbx3 protein? And I'm now going to show you experiments that were done by Una Jin, a postdoctoral fellow in the lab, that include experiments called chromatin IP experiments, where we now add an antibody to Tbx3 and then take out the DNA from the cell that is bound to the Tbx3 protein, comparing to a control, and then sequence the DNA. It's called chromatin IP. And what Una found is that among the targets of Tbx3, there were two genes called E2F7 and E2F8. And E2F7 and E2F8, they're known to be, they were indeed confirmed to be targets of Tbx3 by CHIP qPCR. So what are E2F7 and E2F8? So we've now gone from Wnt to Tbx3. Now we have to go from Tbx3 to E2F7 and E2F8. And what was known from work done by others before, and the citations are here, is that E2F7 and E2F8 are actually inhibitors of the cell cycle. They inhibit cells from going through mitosis. And actually what they are doing, there are in their own right transcription factors that are binding to the promoters of a whole suite of genes that are all implicated in mitosis. So E2F7 and 8, they repress a mitotic program in cells. And these are among the genes that are implicated by themselves in mitosis. One of them is aurora kinase. These are proteins involved in spindle checkpoint, and there are many others. Again, all of these genes are repressed by E2F7 and E2F8. And now you can probably see where this is going. In cells that are out of the cell cycle, that are arrested in the cell cycle, E2F7 and 8, they prohibit cells from going through mitosis. But if a cell is going to be activated by a Wnt, it will now start to the induction of the expression of Tbx3. And Tbx3 is going to repress E2F7 and E2F8. And therefore, it will actually promote cells from going through the cell cycle. Wnt through Tbx3 will actually lead to cells undergoing mitosis. It is an example of a double negative interaction. So how do we know more about this? We actually have done experiments where we have been eliminating Tbx3 from the liver. And I'm not going into the details of the experiments here, but there are these powerful tools in the mice where you can actually eliminate a gene by the combination of a Cre recombinase, and at the same time label the cells with H2B-GFP, the cells that are actually undergoing the elimination of the target gene. And this is what we found. We are labeling cells with axin 2, our Wnt target gene. You see that early in the experiment, there's a nice ring of cells that are being labeled. If you follow those cells over time in a mouse where Tbx3 is still active, it is not deleted yet, you see that the cells are going to divide quite rapidly. But when we now also eliminate Tbx3 from these mice, you see that there are far fewer cells that are being labeled. And it indicates that in the absence of Tbx3, and that is confirmed here in a quantitative way, cells are losing their proliferative capacity. And indeed, if you eliminate Tbx3 from those liver tissues, you see that the expression of E2F7 and 8, so the targets of the Tbx3 protein, are going up. You see that E2F8 RNA by in situ hybridization is now expressed in the liver at much higher levels compared to the control, and also in cells that are normally seeing the activation of Tbx3. When Tbx3 is active, E2F7 and 8 are going up, and therefore the cells are going to be arrested in the cell cycle. So the conclusion is indeed that Tbx3 directly represses E2F7 and 8 in liver tissue, and therefore will allow cells to go through the cell cycle. And I want to reiterate that conclusion again. Wnt regulates mitosis by repressing mitotic inhibitors. And this brings up actually something that I'm really fascinated by, because this is now a common principle of the regulation of a process in a tissue and actually in biology in general. What is happening in the liver, but also in other tissues and in other cells, is that there's a very complex process, such as cell division, and that is being built into the cells. Every cell, in principle, has the ability to divide. That whole process is there. Now, to regulate the process is actually being locked down. And that is happening by the expression of E2F7 and 8 in the liver. These cells are now arrested in the cell cycle until you now start to need cells to be dividing. And what is then happening is that the inhibition of the process is being relieved. And in this case, by a signal, Wnt, working through Tbx3. And if you start to look at other aspects of the cell cycle and many other aspects of growth and regulation of processes in cells, you see quite often a similar principle, where there's a complicated process, it is being locked down until it is actually being relieved. So what are the implications of our findings for cancer? Again, Wnt can activate Tbx3, leading to mitosis. It is indeed found now that there are many aspects of the Wnt pathway that are implicated in cancer. It has been found that there are mutations in axin and APC, and also in beta-catenin in various forms of cancer, including in colon cancer, but also in liver cancer. It is quite prevalent to see mutations in axin and beta-catenin. this work has also been shown that Tbx3 can lead to the promotion of cancer, as you would expect. Overexpression of Tbx3 would in principle activate cells to divide. And that loss of E2F7 and 8 can actually also lead to cancer. So E2F7 and 8 in their own right can actually block cancer because they can block the process of mitosis. It also goes back to the original finding that Harold Farmers and I made many years ago, where we found that the mouse memory tumor virus is indeed something that can activate the expression of Wnt genes in mouse breast cancer through the insertion of the mouse memory tumor proviral DNA adjacent to a gene that at the time was called INT1, and that was later renamed into Wnt1. Now, in the closing minutes of my talk, I'm going to show a little bit about, is this actually general? Is the axis of Wnt Tbx3, is that also playing out in other tissues? And what we found indeed is that Wnt and Tbx3 is activated in many other growth zones in tissues. We also have seen, and other investigators as well, that E2F7 and 8, the genes that are repressed by active Wnt sickling, are widely expressed in tissues where the cells are in mitotic arrest. In one example, are so-called trophoblast giant cells. There's also something that I want to point out at the end that is quite striking if you now start to look at the function of Tbx3. Tbx3 in itself is, as many genes are, a member of a large gene family. And one of them is a gene called brachyury, sometimes simply called T. This is a homologue of Tbx3. These are both so-called T-box proteins. And brachyury, just like Tbx3, is also a repressor. It has the same DNA binding and repressing domains as the Tbx3 protein. And what is very striking about Tbx3, if I now go back to these early embryos across the animal kingdom, where you see the early domains of Wnt expression at the posterior end of Cnidaria or chordates or whatever, is that the T or the brachyury gene has a very similar expression pattern as Wnt itself. And that holds true for the chordates, so a mouse where the tail end, the growing, expanding tail, is active for the expression of the brachyury gene, similar to the expression of Wnt. But in the Cnidaria, again, these are sea anemones and also organisms like Hydra, there's a posterior domain of brachyury that is similar to Wnt. And it has been shown by various investigators that it is indeed the expression of Wnt, the signal, that will induce the expression of brachyury in Cnidaria and in the mouse, suggesting that this axis of a Wnt leading to the expression of a T-box protein and therefore to the proliferation of cells is conserved possibly across the animal kingdom. And those were the experiments and the conclusions of the work. At the end, I want to acknowledge the people in the lab who've been doing these experiments, and that includes Tenny here in a Zoom call, also Una, and then also various other members of the lab. Thank you for your attention. I will be happy to answer questions.
Thank you, Dr. Nusse. That great seminar. I'm going to invite people to have questions to either raise their hand or type them in on the chat. There's a Q&A chat. Don't be shy. I think I'll get the ball rolling if I could myself. One of the things I was curious about as I listened to the evolution of all these pathways and all these things you discussed is what are the drivers of Wnt expression itself?
Yeah, that's indeed a very relevant issue and also something that is hard to study. Typically, what is happening here is that the Wnt's are regulated by enhancer sequences adjacent to the gene. And in the cases where these enhancers have been studied in some more detail, it has been found that the enhancer regions surrounding any wind, and I should explain also there are 19 different wind family members in the genome of almost any organism, that these enhancer regions are wide, are vast, and that there are multiple enhancers. Many of them are tissue specific, so that they can drive the expression of a wind in a particular cell type. And it has been found as well that there are other sickling pathways that actually may lead to the activation of a wind. And in some tissues, it has been found that a sickling pathway that is translating damage to a tissue can lead to the activation of a wind. And this is work that we also has been doing in our own lab, where we found that there is damage to a tissue leading to the activation of the AP1 pathway that will then lead to the activation of wind expression by the binding of AP1 to one of the enhancer sequences next to a wind. So, so in general, again, many enhancers that lead to the activation of wind. And it could also be that there are other sickling pathways leading to the activation of wind in particular cells.
Okay, thank you. Andy Mason, if you'd like to join us, ask your question.
Hi, Rick. Can you hear?
Yes, great, Andy.
Yeah, sorry. The chat is disabled, by the way. Thank you, Professor Nusse. That was a wonderful, wonderful, elegant talk. My question is, have you noted in human pathology any other pathways that would be compatible with mouse mammary tumor virus activity on and above Wnt signaling?
Yeah, in human tissues, that was your question?
So in human diseases, there's some worked on the idea that there's a human beta retrovirus similar to mouse mammary tumor virus. And I'm just wondering if you've seen similar biochemical signatures on and above Wnt signaling.
Well, there's many human tissues where Wnt signaling plays a role also in pathology, but there's no association there with a virus. So one important example is that Wnt signaling is widely implicated in cancer, but also in, let's say, the loss of tissues. And when Wnt signaling is not functional in a situation where it should be functional to maintain the tissue, you get a degenerative disease. So that could be caused by a viral infection, not a homologue of the mouse mammary tumor virus, which is, there's no human homologue of that, but by another cause. So again, both cancer, but also degenerative diseases, that is where Wnt signaling is implicated, either in a way that it is overactivated or in a way that it is not active. I do see some questions in the Q&A.
Yeah, we have several actually.
Yeah.
Just a reminder that you can post your questions in the Q&A, that the chat is not available, as some people point out, but you can post them in the Q&A. Maria asks, is the liver is somewhat unique in that it can regenerate. Does this pathway play a role?
Yes, so the liver, quite famously, you can chop it into pieces, and then the remaining part can grow back into what looks like a normal liver tissue. I just heard a seminar last week of an investigator who showed that in that context, there's also indeed a need for Wnt signaling coming from the blood vessels in the liver, so the central vein in particular. So by that token, yes, Wnt signaling is important for the regeneration of the liver tissue.
Richard Schultz, you had a question. You can join us by unmuting, and if you'd like.
Yeah.
Richard, are you there? you should be able to unmute and ask your question.
Yeah, or we do it from the
lost.
Q&A.
Another question here was a great talk. Could you briefly explain the role of Wnt signaling in osteoarthritis? Thank you.
Yeah, osteoarthritis is an inflammatory disease mostly, but when it comes to the bone, it has been found that Wnt signaling is very important for the proliferation of the osteoprogenitor cells, and that loss of Wnt signaling can lead to osteoporosis, which is not the same as osteoarthritis. I know that, but I mention it anyway. And there's now companies that are trying to activate Wnt signaling in case of osteoporosis to lead to the restoration of bone growth.
Right. Another question here is, what therapeutic cancer options are developed based on the Wnt pathway?
There's several, actually many different companies. On the one hand, they're trying to activate Wnt signaling in the case of a degenerative disease. So there's a number of companies, for example, trying to activate Wnt in the liver in the case of liver degeneration. But the converse is also true. So in the cases of cancer, there's many ways now that people try to interfere with Wnt signaling to block the Wnt activation in cancer. And one of them is that there's a drug that is now developed that would inhibit the production of Wnt itself, that is now in a clinical trial.
Another question here from Paulo is Wnt inhibition a potential way to treat chronic inflammatory diseases, not only rheumatologic disorders, but atherosclerosis as well?
That is a possibility, but I don't know of any data that Wnt is implicated in atherosclerosis, which is mostly a disease where the smooth muscle cells in arteries are over-proliferating, and so it could be that Wnt is implicated there, but there's no data for that yet.
Okay, we have a question here. It was very nice talk. Tbx3 and Wnt signaling you showed in liver. How about heart cells? As we know, Tbx3 and 5 and Wnt also play a role in heart development. My question is that, can we use same principles in HCM disease?
Yeah, that's a possibility. I know of those data. The Tbx5 is involved in heart development. In the adult heart muscle, this is actually one tissue where people have yet to find the possibility that there are proliferative cells in the adult heart, and that those cells could be implicated, that those cells could be activated by Wnt. But for the time being, that evidence is lacking. And that is actually one of the reasons that people now assume that the heart is one of the few tissues that cannot regenerate, and that therefore, heart disease, including as a consequence of atherosclerosis, is not regenerative.
Okay, we have several other questions, but in the interest of time, I think we should probably move on. Thank you very much for those questions, and again, thank you, Dr. Nusse, for a great talk.
Thank you.
We'll move on to our next speaker, I'd like to invite Dr. Taubman, professor in the Department of Cell Biology, to introduce Dr. Paris. Go ahead.
Thanks, Rick. So it's my great pleasure and privilege to introduce our next speaker, Dr. Malik Paris. Dr. Paris first obtained a Bachelor of Medicine and a Bachelor of Surgery degree from the University of Ceylon in Sri Lanka. And subsequently, he moved to the UK and then completed a Doctor of Science degree at Oxford. After completing his DSc, Dr. Paris has held a number of academic positions at prestigious universities in the UK and Sri Lanka before moving to Hong Kong in 1995. So currently he is a professor and the chair of virology in the School of Public Health at the University of Hong Kong. In addition, Dr. Paris is an honorary consultant microbiologist at Queen Mary Hospital, co-director of the WHO H5 reference laboratory and SARS reference laboratories, and co-director of WHO reference laboratories providing confirmatory testing for COVID-19 at the University of Hong Kong. So Dr. Paris has had an outstanding academic career in service in public health, and he's supervised more than 50 graduate and postgraduate level trainees, and he's published over 800 peer-reviewed articles, many in outstanding journals, including Nature, Lancet, New England Journal of Medicine, PNAS, Nature Medicine, and others. He is a fellow of at least 6 professional bodies, including the Royal College of Pathologists in the UK, the Royal College of Physicians, and the Hong Kong Academy of Medicine, and he holds multiple honorary doctorates and too many other awards to mention in this venue. In 2021, Dr. Paris was awarded the John Dirks Canada Gairdner Global Health Award for his work, which led to understanding the origins and options for control of newly infectious disease outbreaks in Asia, notably zoonotic influenza and severe acute respiratory syndrome, also known as SARS. So together with Dr. Guan Yi, also at the University of Hong Kong, he initiated seminal studies on the underlying causes of H5 virus pathogenicity, the evolution of the H5n1 virus. They also developed a highly effective monitoring and surveillance program for avian and swine influenza virus strains. Doctors Guan and Paris established that live poultry markets in southern China and Hong Kong were the source of the virus spreading to humans, which in some cases exhibited up to 60% lethality. Their subsequent work established new protocols for periodic live poultry market closures, emptying these markets of poultry overnight to reduce virus amplification in these markets, and the appropriate use of poultry vaccines to protect both poultry and people in Hong Kong from H5n1 infections. They also made major contributions towards understanding the emergence, transmission, epidemiology, and pathogenesis of highly pathogenic avian influenzas, including H5n1, H9N2, H6n1, H7n9, and others, and have provided evidence-based options for control of avian influenza viruses in Asia. In 2003, Dr. Paris led the team that first identified the responsible for the SARS epidemic, which is known as the SARS-CoV-1 coronavirus, also elucidating its pathogenesis, transmission, and quickly developed a diagnostic test for the virus, which was then shared internationally. The isolation and characterization of the causative agent of SARS and quick development of a diagnostic test directly influenced public health policy to effectively monitor and control the spread of the disease. Dr. Paris and Guan's comprehensive strategies for surveillance, diagnosis, and control of emerging infectious disease outbreaks was again proven important for mitigating the morbidity and mortality caused by the 2009 swine flu pandemic, the outbreak of Middle East respiratory syndrome or MERS, and now the current COVID-19 pandemic. Dr. Paris's talk this morning is entitled From Pasteur to SARS and Beyond, A Personal Journey. Welcome, Dr. Paris.
Thank you. And thank you very much, Dr. Hubman, for that kind introduction. And I'd like to thank the University of Alberta for organizing this meeting, the Minds That Matter. And of course, my thanks to the John Dirks Canada Gairdner Foundation for awarding Guani and myself the 2021 Global Health Award, which of course is not just for the two of us, but really is a recognition of our whole team. If I can try to share my screen. Yes. Yes, so I have entitled my presentation From Pasteur to SARS and Beyond, A Personal Journey. So when I was, so what I will try to do is give you a couple of overlapping stories, and are not necessarily sequential in time. I'll have to come back in time because they are overlapping in time. When I was 14 years of age in Sri Lanka, I read a book on the life of Louis Pasteur, and I was really fascinated by this scientist. He not only made very important discoveries in organic chemistry, but he established the germ theory of disease, the scientific basis of vaccination, translating this basic science to application, and quite importantly, disseminating this knowledge worldwide through the establishment of Pasteur Network laboratories. And as I was to learn later, it was one of his students, Alexandra Yersin, who came to Hong Kong in 1894 and discovered the cause of the plague. And what you see there is his temporary laboratory, which in fact, although it's not there now, it was not far from where my laboratory today is situated. And as life would have it, in 2009, I had the honor to become one of the directors of the International Pasteur Laboratory Networks laboratories in Hong Kong. So let me go back to the early beginnings in Sri Lanka. As was mentioned, I did my undergraduate training in medicine, but right through, I wanted to become a researcher. And in this, I was really inspired by the then professor of microbiology, Chabi Arasakulratna, at my alma mater, the University of Peradeniya, because he really showed that even in resource-limited settings, that you could do good science, whereas most other academics, unfortunately in Sri Lanka, were very dismissive of the capacity or the ability to carry out science in a place like Sri Lanka. As was mentioned, I did my doctoral training at the University of Oxford. I returned then to the University of Peradeniya to try to set up a cell culture and virology laboratory there. Now, this was quite challenging because even simple things like the ultra pure water that you need for cell culture was not available. I had to traipse across city to a veterinary lab, which was a vaccine laboratory, and regularly bring in the ultra pure water from there. As many of you will know, one of the basic items of equipment for cell culture is a carbon dioxide incubator to maintain a 5% atmosphere of carbon dioxide. But this, at the initial stages, was not available. But luckily, I had seen in one of the labs that I worked, somebody using this trick of using transparent plastic boxes, and you put in a small container a bit of sodium bicarbonate, add in some dilute acetic acid, and immediately seal the box. And you can, by getting the proportions right, you can generate an atmosphere of carbon dioxide in order to do those experiments. So that is how I really got this lab up and running. In due course, of course, I was able to get some of these equipment through grants from the Wellcome Trust and NIH. We made some worthwhile research contributions, including making the first monoclonal antibodies to Plasmodium vivax in collaboration with a parasitologist colleague, and using those to identify the targets of blocking transmission of the Plasmodium vivax parasite, and studies on mosquito-borne viruses. But what I want to talk about in particular is this experience amongst some of this 6-year period that I was in Sri Lanka. The Mahaveli River, the longest river in the country, was being dammed for hydropower and for irrigation. And so this was leading to a huge ecological change in a fairly large part of the country. together with an entomologist colleague and myself, we were wondering whether this ecological change might have impact on emerging mosquito-borne or tick-borne virus infection. So we set up a study site at the stage of the forested stage of development, and basically stayed there right through the phases of development of clearing of the forest, irrigation, introduction of agriculture, and human settlement. And we were expecting, of course, some emerging outbreak to occur there. But as usual, what happened was there was an outbreak of a mosquito-borne disease, but that occurred in a different part of the country, which was very stable, had not undergone this type of ecological change. But nevertheless, since we had all the tools available, we went to investigate that outbreak, and we identified the mosquitoes that carry the virus. We characterized the human outbreak, characterized the virus itself. So the question was, why this particular outbreak now? Just to give you the perspective, Japanese encephalitis had been reported in Sri Lanka, but the whole country had maybe 10 to 12 cases a year. And now we had 500 cases in a single hospital in 3 months. So what exactly had changed? So like I said, we looked at the mosquitoes, we looked at the virus, we looked at the environment. And in fact, what proved to be the driver of this outbreak was a very well-meaning intervention by the local provincial government to introduce pig breeding as a way to supplement the economy of the poor peasant farmer. So every farmer was given free of charge 10 or 12 pigs to have in his backyard for protein as well as for sale. So the pig is an amplifier host for Japanese encephalitis. So in a situation where the virus was present in wild birds, the environment was ideal for the mosquito vector, which was present in abundance. This very well-meaning introduction was what led the spark to this huge outbreak. Now, we then went on to investigate what other parts of the country might be at risk of Japanese encephalitis, and what parts of the country might be safe for pig husbandry. And and this led us to conclude that there were 2 areas that were at high risk. And one, in fact, was exactly that area that we were studying undergoing rapid environmental change. And in fact, as part of that development program, there were, in addition to providing irrigation, there was also an animal husbandry component, and pigs were part of that component as well. But with the evidence from the previous outbreak, we were able to explain to them that that would be a bad idea. And luckily, we were able to stop at least introduction of pigs, although other livestock would be safe. But equally, we found other areas of the country, particularly at high elevations in the hill country, which were, from a vector-borne point of view, quite safe for animal husbandry. So this really taught me an important lesson. And of course, today, this is what is called One Health. In other words, optimal health of people, animals, and the environment. And the fact that we really have to look at these problems from not just from a human health angle, but together with animal health and environmental sciences as well. Now, fast forward almost a decade, I then took up an appointment at the University of Hong Kong. And one of my tasks was to set up a diagnostic virology lab for the Queen Mary Hospital in Hong Kong. And within 2 years of taking on that job, we were then faced with the avian flu H5n1 outbreak in Hong Kong. This was a highly pathogenic virus of poultry that started to spill over to humans. We had 18 cases and 6 deaths. And the source of human infection was identified to be these live poultry markets and Hong Kong-wide depopulation of poultry, successfully brought that outbreak under control. After that initial outbreak was brought under control, I was interested to look at whether the virus is persisting in the wider environment. And that is where I joined my co-awardee, Yi-Guan, very much under the mentorship of Robert Webster at St. Jude Children's Research Hospital in Memphis. He, as some of you might know, is probably regarded as the father of modern influenza virology, and he was immensely instrumental in both of our careers, Guani's and myself. To cut a long story short, we could see evidence of the precursor virus, that H5 virus, in geese, and then we could see it evolving. Influenza viruses have 8 gene segments, so you can see them swapping gene segments, adapting from geese to ducks and then back to chicken. And we were clearly very much on alert to problems that might arise. As part of this response, we had set up an avian influenza task force, which included participants from the Department of Health, Department of Agriculture, the Department of Food and Environmental Hygiene, and the universities. So again, this was One Health in action before the concept of One Health became as popular as it is right now. Through these studies, we were able to show that what was going on is that poultry were coming in from many different farms. And of course, at that time, 90% of poultry sold in, live poultry sold in Hong Kong was imported from mainland China. They came into one large wholesale poultry market. And from there, it was distributed to a number of small retail poultry markets, such as what you see here. And what we were able to show is that these small retail poultry markets were an ideal venue for the virus to persist and thrive. So, for example, one of the proofs of this was that when we, well, first and foremost, you could clean these poultry markets as much as you like, but you will not get rid of the virus because, after all, the virus is in the poultry themselves. So the only way to interrupt transmission was to empty the market completely of poultry. And here you see one of those studies that was done, 8 poultry markets. Before closing the market for one day, 6 of these markets had evidence of, this is not H5n1, but another avian influenza virus, which is less pathogenic, but we use it as an indicator of avian influenza virus activity. You can see that there was evidence of virus activity. Day or two after closure and reopening, it had dropped almost to 0. But by the next month, it is up again. Close the market for a day, it is down again, up again, down again. So proving this concept that the virus enters infrequently, but once it enters, it remains there. And then we also were able to show that outbreaks in poultry farms were also related to these live poultry markets by people moving cages and the people themselves carrying the virus back into these poultry farms. So with this knowledge, we were able to introduce a whole series of interventions, including rest days, where one day of the month or 2 days for the month, the poultry market was closed. And then later on, when the market was empty of poultry overnight. So in the morning, poultry come in, they are sold, but they're emptied overnight. And you can see the very dramatic reduction of virus activity in these markets with these fairly simple management interventions. With this surveillance and the awareness and the actions that were taken, and I'm giving you only just a few of the interventions that were taken, there have been no further cases of severe avian influenza in humans in Hong Kong since 1997. However, in 2004, country after country, region after region, awoke to the fact that they had this highly pathogenic H5n1 virus within their borders and spilling over to cause human disease. And I think the lesson there is that proactive surveillance, proactive interventions based on surveillance and evidence-based interventions can protect from subsequent much worse outcomes compared to if you don't take these measures. Later on, we were also, my colleague Yen Huiling, working in collaboration with the colleagues in southern China, because there was some evidence that some of the patients who developed avian flu disease did not actually touch poultry or even go into the live poultry market. So we wondered whether the virus was present in the air. So we did air sampling in these markets, so we could detect live virus in air samples and identify this bit of machinery called a defeathering machine. This is after the poultry are killed, they are put into this with some water and spun around to remove the feathers. And as you can imagine, this generates a lot of aerosol, and this was the area of highest risk for detecting virus in the air. And in collaboration with colleagues in our Department of Engineering, we've been able to do some simple modifications to this apparatus to reduce the risk of aerosol spreading. So this is airborne droplets coming out in the original version, and this is the modified version. So these are some of the measures that we have been taking to reduce the risk of avian influenza. Now, another understanding that Guani and I got to through these studies of virus evolution, with, for example, studying first the emergence of the more recent H5n1 virus, but it was also true of H5n1 and H10N8 and other concerning avian influenza viruses, was that 6 of the gene segments, if you remember, I told you that influenza viruses have 8 gene segments. 6 of these gene segments come from a very common poultry virus, a chicken virus called H5n2. This picks up the H and N gene segments from wild bird viruses, and that comes from wild birds to chickens through domestic ducks. So this is the pathway, and the fact of the matter is that the marketing system at the moment mixes aquatic poultry like ducks and geese together with chickens, and that is what allows these novel viruses to emerge. So separation of the marketing chain between aquatic and terrestrial birds would also be a generic measure that, in theory, could reduce the emergence of future zoonotic influenza viruses. Now, one of the other things that I was interested in about avian influenza was the severity of the disease. Avian influenza H5n1 was associated with a mortality of over 50%. And we had shown that in vitro, the host response, particularly the innate immune response with H5n1 influenza, was very different to what we saw with seasonal influenza. So we believe that the pathogenesis was partially linked to this dysregulation of innate immunity. We have subsequently, this work done by one of my colleagues, Michael Chan, and myself, we have set up an experimental model to mimic what goes on in the air spaces, the alveolar spaces of the lung. And what we have demonstrated is that when a virus like H5n1 infects these alveolar epithelial cells, they release soluble mediators that then feed back to downregulate these sodium and chloride transporter channels that are found on the alveolar epithelial cells. Now, these channels are essentially pumps that remove fluid accumulating into the alveolus. They pump it out so that the alveolus is kept free of fluid and able to do its job of air exchange. If you block these pumps, essentially, you accumulate fluid in the alveolar spaces, and that is what leads to the acute lung injury and severe pneumonia that you see in these virus infections. We then went on to show that this damage can be reversed with certain interventions, such as mesenchymal stromal cell therapy. These are the treated mice. You can see the lungs looking much better than the untreated mice here. And now we are using these models to explore options for other small molecule therapies that can reverse this fluid accumulation. let me now move back in time to 2003. That was the time that we heard about this unusual pneumonia that was to be called SARS, taking place in Guangdong in southern China. In Hong Kong, we are at the border of Guangdong. So it was obvious that the virus would spill over to Hong Kong. We set up surveillance together with the hospital authority. We were able to isolate a suspected virus, which we showed to be associated with this disease. We obtained partial sequence information, which showed that it was a novel coronavirus, not identical to any of the known animal or human coronaviruses known to humans before. And then with this sequence information, we were also able to set up diagnostic tests. So within a week of isolating the virus and sequencing it, we had developed diagnostic tests, which we rolled out progressively in our hospital system. Now, this, of course, is not what we would do normally. We would have a much longer period of assessment and evaluation of a diagnostic test, but this was an emergency, and essentially, we had to build the ship as it sailed, and luckily, that job was done effectively, and the tests worked perfectly well. Using these diagnostic tests, we were able to characterize a number of features of the disease. So we found that the virus was not only present in the respiratory tract, it was also present in the stool. And indeed, some of the patients did have quite a significant diarrhea as part of the presentation. And at least in one example in Hong Kong, there was a large outbreak that was associated with fecal aerosolization and contamination. There was another very unusual feature to this disease, in that if you look at the quantity of virus in the upper respiratory tract of these patients, in the early phase of infection, the amount of virus in the upper respiratory tract was very low. It was at the end of the first week that the viral load increased dramatically. As you can see here, this is a logarithmic scale. So what this meant was that it's likely that early in the infection, patients were not so infectious. And indeed, that turned out to be the case. The epidemiological data also suggested that transmission was much less likely in the first 4 or 5 days of illness. Now, there was another interesting and fortuitous finding in the case of SARS. What you see here is a very majority of common respiratory viruses like influenza. You see that most of the infections are very mild and only a small proportion of them are severe. In the case of SARS, the vast majority of these infections were very severe. And this, of course, helped to identify the cases and diagnose them and get them out into hospitals so that the chain of transmission in the community could be broken. And that is how the outbreak of SARS was contained globally. And so that it was March when we first, the infection, or February when it spilled over in Hong Kong, March when we identified the virus. But by July, it was possible for WHO to state that all known chains of person-to-person transmission of SARS may now have been broken. Now, other viruses such as influenza have a very different profile of viral load, so that patients are infectious right from the time of onset of symptoms or even before. And in fact, when COVID-19 emerged, we collaborated with colleagues in Guangdong very early on, and by February of last year, we had reported that the profile of viral load of COVID-19 resembles influenza rather than SARS, so that the measures that were able to contain SARS were very unlikely to be able to contain COVID-19. And sadly, that proved to be the case. Now, part of this SARS response was a really unique and great experience coordinated by WHO when about 12 of the laboratories worldwide who were dealing with this problem were linked up every night, every day, it was nighttime for us, with teleconferences. And we were sharing the information that we were gathering in real time. And indeed, some of the people I got to know during this experience was the late Donald Lowe, who was the head of microbiology at Mount Sinai Hospital in Toronto, and Theresa Tan, who I believe is now the chief medical officer in Canada. The next question, of course, was where did SARS come from? It was clear it was a completely new virus. It was very likely to come from animals to humans. My colleague Guani then followed up a number of epidemiological clues to go and test these large game animal markets that you find in southern China. And he found evidence of SARS-like viruses there. And it was that information that subsequently led the Guangdong authorities to close these game animal markets because the virus was persisting there even after the human outbreak had been brought to a halt. And I'm absolutely sure that that intervention prevented the reemergence of this virus from this source. Subsequently, it was shown that the natural reservoir of the virus are these small Chinese horseshoe bats. And the exact identification of the closest precursor to the SARS 2003 virus took almost 10 years, as you can see here. Now, you might ask why SARS did not emerge before. The fact of the matter is that this culture of eating wild game animal meat has been a traditional cultural habit going back centuries. But the large game animal markets where hundreds and thousands of animals were brought together was a recent phenomenon as part of the affluence taking place in southern China and the demand creating a supply. And then, of course, air travel, which has increased so much, that means that any outbreak anywhere in the world can be everywhere within a matter of days. So many of the changes we ourselves as humans introduce are facilitating the emergence and spread of some of these viral diseases. In the case of COVID-19, I will just highlight two of the contributions we made. Very early on, we developed the diagnostic tests for COVID-19, and it was one of the first diagnostic tests to be on the WHO website. And we have on request distributed reagents for these tests to over 170 laboratories in 77 countries. And a number of countries made their first detection of COVID-19 using these tests. This was, of course, before commercial tests became available. Another important finding that was this was work that we were doing before SARS, because we were very interested in transmission of respiratory viruses, particularly influenza. But some of the patients we were studying were other coronaviruses, not SARS, not COVID-19, not SARS coronavirus 2, but other coronaviruses. And what we could see in these other coronavirus infections was that infected patients were shedding these viruses in the air through, these are air sampling studies, and both large droplets and small droplets, aerosols, were carrying these infectious viruses. And even more importantly, when the same patient wore a surgical mask, we could see that the release of these airborne particles was largely blocked. So this was one of the factors, or particularly once it became clear that patients with COVID-19 were being infectious even before they developed symptoms, this data became important as one of the evidence to argue for universal masking of the general population, even if they were apparently healthy. Now, before I close, I just want to turn to another coronavirus disease, and that is MERS coronavirus. You may have heard this is the Middle East respiratory syndrome. There have been repeated outbreaks over the last number of years since it was first reported in 2012, some outbreaks of over 100 or 200 cases, and one outbreak from one infected person in South Korea leading to 186 cases. Now, one of the things that fascinated our group was the fact that, well, dromedary camels are known to be the source of human infection, but most of the dromedary camels in the global worldwide are found in Africa, not in Saudi Arabia, whereas all the human cases that originate from camels to humans occur in the Arabian Peninsula, although travel-associated cases may be found elsewhere. So why was no MERS cases being reported from Africa? So to cut a long story short, we have done studies in multiple countries in Africa. We showed that the amount of virus in camels is no different to that in the Arabian Peninsula. But when you look at antibodies in camel workers, we don't have evidence of infection. We don't see evidence of antibodies as evidence of infection. However, when we look at T cells, which is the other arm of the immune response, here you can see the data showing that CD4, CD8 T cells, these are positive control patients from Saudi Arabia. These are negative control patients. They're very clear. And now we have camel workers in an abattoir in Nigeria, workers in that abattoir not directly working with camels, and controls from the city, adjacent city. And you can see that the camel workers, about 30% of them, actually have very specific signals for MERS coronavirus T cell responses. So, and we have in the paper shown why we are confident that these responses are specific. Phylogenetically, we can see that the viruses in Africa are distinct from the viruses in Saudi Arabia or the Arabian Peninsula. And we can show that the viruses in Africa have lower replication competence in the human bronchus and in the human lung compared to those viruses in the Arabian Peninsula. Using pseudoviruses, pseudotyped with the spike of, say, the Saudi virus compared to the African virus, we can see virus entry is reduced. So it appears that, first and foremost, that human infection is taking place in Africa, but is largely unrecognized and ignored. And we really need enhanced surveillance of MERS coronavirus at the animal-human interface in Africa, in the camel herding regions in Africa. So if I can summarize, I've tried to show the importance of the One Health approach, the fact that we need to look at these emerging infectious disease problems from the human, animal, and environmental health point of view. The importance of surveillance and risk assessment. I hope I showed that with the examples we used for avian flu, for SARS, and for MERS. Unfortunately, now there is a tendency to regard this type of work, surveillance at the animal-human interface and risk assessment of these viruses, as dangerous science. I submit that this is not dangerous science. It is essential for pandemic preparedness and for identifying measures for risk reduction. I fear future pandemics are inevitable. COVID-19 is not the most severe pandemic conceivable, and I think we really need to learn the lessons from COVID-19 to be better prepared for the future. And I think I would like to end by going beyond pandemics. I think COVID-19 has highlighted the limits of human hubris, and I'm struck by the quotation by René Dubos, a French microbiologist and philosopher, back in the 1950s, when at a time when a lot of people thought that antibiotics and vaccines were essentially controlling infectious diseases. He said that at some unpredictable time and in some unforeseeable manner, nature will strike back. And what we see with COVID-19 is nature striking back. And indeed, it is not just with pandemics. I think nature is striking back in regard to climate change, environmental degradation, environmental pollution, And as a human species, we are running the risk of rupturing the limits of planetary sustainability. And I think I will end with this thought that while, of course, we are concerned about pandemics and COVID-19 now, and hopefully how we can deal with pandemics, deal better with pandemics in the future, this is a problem beyond pandemics. And I think we really have to take cognizance of arranging our societies in a more sustainable manner. So with that, I will end just with my acknowledgements. This is a photograph taken soon after SARS, and it has a number of the key people who contributed to this. This, of course, is Guani, the co-awardee, but Leo Poon, K.H. Chan, John Nichols, who did electron microscopy, Leo Poon, who got the initial partial sequence, K.Y. Yuen, who did a lot of the clinical work. And of course, I again want to emphasize the mentorship of Robert Webster throughout my latter part of my career. Gabriel Long, our dean and previously head of the School of Public Health, and of course many others that I've had the good fortune to collaborate with. And with that, I will end. Thank you very much for your attention.
Thank you very much, Dr. Paris, for that very exciting presentation. There are a couple of questions on the chat. I'll start with those. The first one was, if the profile of viral load is identical for COVID to influenza, despite having greater genetic closeness to SARS, I was wondering if there is a much higher selective pressure for COVID virus in terms of pathogenesis. What is your opinion?
Well, the similarity in the viral load profile has implications for transmission, because what it means is that patients are transmitting even before they develop symptoms, because you certainly have high viral loads even before they have symptoms, and in the first few days after onset of symptoms. So that is the primary problem that we face, and unlike SARS, where we had this window where if you could catch them and get them into isolation, we could break the chain of transmission. Now, there is a bit of a mystery there, because after all, as far as is known at the moment, the receptor for SARS-1 and SARS-2 is the same. It's the same ACE2 receptor. So one might ask why there is such a big difference in this profile of viral load kinetics in the two diseases. And I don't think we have the full answer to that. Co-receptors may be an important part of that, but certainly the consequences for public health are quite dramatic.
Another question here. Do you have any comments about the value and or risk of gain of function studies?
So I think this, of course, is a hot topic at the moment. Now, there's no question that gain of function studies have to be regulated and considered very carefully before they're done. And of course, you have to also consider what exactly is meant by gain of function. There are a lot of studies in virology that involve manipulating genes of the virus itself, or maybe swapping genes from one variant of the virus to another variant of the virus. These types of studies would not be gain of function, because we are dealing with things that are already present in the environment and out in nature anyway. Now, of course, there are other studies, and this particularly came to the fore with the H5n1 studies that were done back in 2012, when the aim of the study was to try to see if the H5n1 virus could be transmitted by the airborne route in an experimental model system. And that rightly led to a pause, a lot of concern, and a pause of those studies until a structure was put into place to assess what was, first and foremost, assess whether a study was worthwhile to do, whether there was a rationale, whether there was the benefit that could be obtained would outweigh the risks. And secondly, whether such a study could be safely done and under what conditions it could be done. And some of these studies have been allowed to proceed. Now, I think this type of studies are important, but without a doubt, they do need to be considered very carefully and regulated very carefully as well. And the proper, appropriate levels of containment should be used for these studies.
Another question, you mentioned MSC therapy in animal studies. There have been hundreds of clinical trials now being done for MSCs for COVID-19. Please comment, is your HKU, Hong Kong University, or your team involved in any of these trials?
No, we ourselves are not involved in any of these trials. I'm aware of the work that's going on with COVID-19, but also before that in other contexts of acute respiratory distress syndrome. What we are, well, because it is somewhat challenging to do these mesenchymal stem cell therapy interventions, what we are focusing on is to try to identify small molecules that would do the same job of reversing the block of these transporter proteins that we observed in our experimental model.
And the last question on the Q&A site is from Dr. Lauren Terrell, who's a longtime supporter of this event. Do you see as the future of COVID, what do you, see as the future of COVID-19, and will it ever disappear or remain a threat long-term?
Well, I mean, the virus itself would certainly remain. I mean, I can't see the virus disappearing. So it will become another one of those endemic respiratory viruses. I mean, keep in mind that over the last maybe 500 years, we have had at least 2 other coronaviruses that have jumped from animals to humans. The 2 common cold coronaviruses, 229E and OC43, we now realize came from cattle and maybe from camels, in fact, or from bats to humans over the last 400-500 years. And now, of course, they're widespread. So COVID-19 will become like that. I think the challenge for us is to be able to learn to live with this, so that the more severe parts of disease can be modulated through vaccination and then ultimately by immunity. And then hopefully with more therapeutic agents coming online, we will be able to continue to coexist with this virus, but I don't see it disappearing.
Okay, I don't see any more questions at this point. I will thank you again for those responses, and I'd like to end by thanking both speakers, Dr. Nusse and Paris, for their joining us virtually in Edmonton, and for their outstanding presentations and their response to our queries. With that, I would conclude today's symposium, and hopefully everyone enjoyed it. Thank you for coming, and have a great day.
Thank you.